Welcome to the deep dive. Today we're looking at some really advanced biomedical engineering concepts.
That's right, we're drawing on cutting-edge research, including key work from places like the Ravi Radhakrishnan lab.
And we're exploring how engineers are tackling one of cancer's biggest hurdles.
Yeah,
Its incredible complexity, its heterogeneity.
exactly, by looking at, cellular messaging, physical forces, even computational modeling.
So the mission for this deep dive.
It's really to show how analyzing these tiny microscopic signals, these mechanical cues, how that's pushing us way past old methods like static biopsies
Or something much more dynamic.
towards patient-specific cancer digital twins. We're talking real precision medicine here, based on insights from top journals like Nature Biomedical Engineering.
Okay, let's start with the cell communication part. Extracellular vesicles, EVs, and specifically exosomes. Tiny things. Weren't they thought of as just like cellular garbage disposal for a while?
They were, but now we understand they're absolutely critical. Think of them as encrypted messages between cells, really central to personalized medicine.
How so? How do they help with being patient-specific?
Well, they're key for molecular profiling of the disease at, pretty much any stage. They carry this complex molecular cargo,
Cargo like what?
signaling proteins, bits of RNA, DNA, things that can literally reprogram the cells that receive them.
Wow, okay. So they can drive metastasis or drug resistance.
Precisely. And by sampling this cargo non-invasively, maybe from blood, we get this real-time snapshot of what the tumor is actually doing, its operational state.
So we've got these messengers, the EVs, but what influences the message inside them? You mentioned physical forces.
Yes, mechanobiology. This is fascinating. It's the link between physical forces and all this EV activity.
Because the tumor environment itself changes physically, right? The tumor microenvironment, TME.
Drastically. It often becomes much, much stiffer. That's due to changes in the extracellular matrix, the ECM, the scaffolding around the cells.
Okay, so it's stiffer. But how does that physical property, talk to the biology? How does it change the messages?
That's the crucial question. Research shows a really important feedback loop here. The mechanical cues, like that ECM stiffness, they don't just sit there, they actively boost cancer-causing signals.
So stiffness makes cancer signals stronger.
Yes, and it directly modulates EV formation, how many are made, what specific cargo gets packed inside, and how readily they're secreted.
Wait, hang on. So the actual physical hardness of the tumor environment tells the cancer cells to send out more aggressive messages, perhaps more resistance signals, via these exosomes.
That's exactly what studies show. For instance, take certain cancer cells, culture them on a stiff ECM versus a soft one.
Yeah.
On the stiff matrix, they pump out way more exosomes, and the EVs they release, they make recipient cells move more, become more modal.
So you're directly linking the patient's specific physical tumor environment to their specific pattern of cellular communication. That's a huge piece of the puzzle.
It's critical for prediction. You can't ignore the physics.
Okay, this complex picture molecular messages, influenced by physical environments, it feels like it demands a sophisticated way to put it all together. Which brings us to cancer digital twins. Verifiable cancer digital twins, VCDTs.
Exactly, that's the goal.
Define that for us. What is a VCDT, really? More than just a computer model.
Think of it maybe like a high-fidelity flight simulator, but for a specific patient's tumor. It's a virtual construct, yes, but it mimics the tumor's structure, its context, its likely behavior over time.
And it needs serious computational power.
Oh, absolutely. It has to use advanced mathematical models, things like multiphysics, agent-based modeling, sophisticated frameworks.
To integrate all this diverse patient data we've been talking about. The stiffness, the exosome contents, genomics, proteomics, tissue images.
All of it. The aim is to predict how the tissue as a whole will respond. It's not static. The VCDT is meant to be dynamically updated with the patient's ongoing data.
So clinicians could
What?
test treatments on the twin first.
That's the idea. Instead of trying treatments sequentially on the patient, you run virtual trials on their digital twin. Test hundreds of strategies, perhaps. Predict potential failures. Optimize combinations, timing, dosage, all before giving the actual first dose.
That's a paradigm shift. Integrating everything from nanoscale mechanobiology affecting tiny exosomes all the way up to these large-scale VCDTs. It really could transform targeted therapy.
It could. But the key word you mentioned earlier is verifiable. Trustworthiness is paramount.
Because the tumor and the patient are always changing.
Constantly evolving. So building a reliable digital twin isn't a one-off task. It's not a static model. It demands this continuous cycle of VVUQ verification, validation, and uncertainty quantification.
VVUQ. Making sure the twin stays accurate.
Essentially, yes. It's about rigorously testing the model, comparing its predictions to real-world outcomes, and understanding the boundaries of its predictive power, putting confidence levels on those virtual test results.
So the twin remains a dynamic, trustworthy replica of a changing reality.
That's the absolute necessity for it to be clinically useful.
Which leaves us with a final thought for you, the listener. If we get really good at this, if a VCDT accurately predicts, say, drug resistance months before it's clinically obvious, what are the challenges, ethical or practical, that arise then? Something to consider.